feeling, no, let me put it up. So you want to see what patterns. And let me tell you, there's a difference between an amino acid and an amino acid residue. An amino acid is just standing alone. A residue means an amino acid that is attached to another amino acid with the hope of a polypeptide and a protein. Please note. So when we say pairwise, meaning two, multiple, more than two. So pairwise, you have two sequences that are matched up with each other, and you want to see how they align. If you bring in your knowledge from microbial genetics, you'll see that there are changes that can occur in sequences. And like we like to say about the type of changes we're talking about, if we talk about a heritable change, that is mutation. We typically can identify areas where sequences have remained the same, similar, conserved. We can also identify regions where they vary. Now, going forward, simply conservation would mean that there is evolutionary relatedness between two organisms. If there are variations, the changes can be due to evolution because of things like mutation. And what types of mutations can occur? Indels, insertions, and deletions, or we can just have substitution. Those are the key things I want you to know. What about sequence similarity? In sequence similarity, we refer to the fact that the two sequences belong to the same protein family. So they are similar. In their similarity, they would be similar in their structure. They will be similar also in their function. Please note that. Now, there are two things that can happen. You can have two sequences that are aligned and you want to tell what type of evolutionary relationships that they have. So you can align two sequences and they may not be identical, right? So you may find one region or one sequence. It may look like another region in another sequence, but then one half looks like one sequence. So things happen. So we talk about even divergence. So different things happen about resemblance. You see these things in your structure. You see, you pull up one protein, you blast it, or you go to HH and you say, the one half of it looks like something in Abby's shoes, another half looks like something in another phage genome. Sequence homology just tells us how, to what extent sequences are similar. It could be qualitative, homologous, or non-homologous. And when we talk about homology, we're just saying that the sequences must have come from a common ancestor, meaning they have a similar evolutionary origin and it's simply based on sequence alignment. So that's the qualitative part. The quantitative part, we can actually measure it. Yeah, you actually see there's a number for it. When you download your BLAST data, you see that these are noted in percentages. Now, is similarity due to random chance? Well, a nucleotide sequence is one of 4 bases ATGC. A protein sequence is one of 20 amino acids. So if we think about the length of the sequence, a shorter sequence may occur due to random chance, but it's less likely that a longer sequence will just be occurring due to chance. And we have seen annotations. I don't know how many. We've made decisions. What does it look like, etc. So sequence similarity and sequence identity. These are the considerations when you think about your BLAST that are taken note of. And so remember how I talk about nucleotides? If you're talking about nucleotides, there's an idea, there's a way to look at it. If you're talking about proteins, there's also a different way to look at it. In sequence similarity, when we think about proteins, we're thinking about the aligned residues. Those aligned residues are what? Amino acids that could be substituted for each other because they have the same physicochemical parameters. Aspartic acid, glutamic acid, they have similar physicochemical parameters. So you could think that way. Then sequence identity. Now you are talking about the percentage of matches of the same amino acids between the aligned sequences. Oh, I saw aspartic acid here, I saw aspartic acid. I saw glutamine here, I saw glutamine. You are just referring to the match. So, calculating sequence similarity and identity in proteins. I remember in Kenao Suji, I think this was his DNA master file at the time, and I apologize, this could have been key one. If people had asked you for screenshots or you had submitted your files to me, it could have been any of yours. So now let's look at gene 1, and I'm very happy about this display. For those of us who have not familiarized ourselves with DNA master, this is a very, very critical slide I need you to look at. Overview, features. This table is talking about features. So when a gene, we have set up the numbering to go this way, and in our style, green means forward, and we've set red for reverse genes. Now, the cursor showing this black arrow means that we are referring to gene 1. And remember, if we talk about sequences, we say DNA has what polarity? It goes 5' to 3' for forward genes and 3' to 5' for reverse genes. Now, if you look here carefully, this is the description. And you can see that it's pulled up HNH endonuclease. And it's showing whether, I forget whether it was Zafira or what that similar phage was, but it's pulled a similar hit or many similar hits, and they're on display. Now, I want you to look down with me. It tells you length, it tells you maximum score, it tells you a bit score, it tells you an E value, it tells you the length, percent aligned, 87.8%. So when the two sequences matched up, it did not align 100%, but out of the 87.9%, the query 1 to 86 or 1 to 96, they pulled out this exact, and they had an identity of 98.84, similarity 98.84. So if you look at this example, they had matching identity and matching similarity. Okay, so method 1, how are we going to calculate the sequence similarity? What we do is that we use the overall sequence lengths of both sequences. So this is the formula. So LS times 2 over LA plus LB times 100. And of course, observe the parenthesis. So where S is the percentage of similarity, LS is the number of aligned residues with similar characteristics. LA is the length of sequence 1. LB is the length of sequence 2. Now, if you could get the sequence from alignment by the side of HSP. So when you get this slide, I want you to practice it with any protein of your choice. Once you follow these steps, you'll be able to arrive at this same spot. So click on export, select all the sequence displayed. You can copy it to a Word document, PowerPoint, Excel. You will look for the areas where, this was from Phage Peterson, where it differs from the target, where your query differs, okay? The difference is highlighted in red, and there is just one. I wonder why the red didn't show here. So I want you to look at all of these and tell me where they are different. I want to see who can tell me where the difference is. There's only one difference. Who knows where the difference is?
The G and E, man. G and
Pardon?
E.
What?
G and
The
E.
G and E on one.
The G and
G.
E on
Yes.
one. Will you say they are different? The G and E. Every other thing is the same.
Yes.
Are you sure?
Yes, ma'am.
What is G?
Glycine.
And what is E?
Glutamine.
Vitamin.
Glutamine. Are they in the same
Glutamine.
group? Do they have the same physical chemical parameters? So you got it. So you have G, and that is a polar amino acid, and your target has an E. Gluta is an acidic amino acid. But remember that your G for glycine is different from your glutamic, which is acidic. So right here, there is a difference. They do not have the same physicochemical parameters. Okay? So in all that sequence, they were only different where in one spot. So let's go back. Right here, okay? We looked at the percent aligned. We had a query, that's the length. We had a target, that's the length. This is what we plug in. And then we calculate based on our LS. And this is what we get. So I can give you this very easily in the exam. So you need to know this. It's inevitably you are going to find it as a question. But please, does everybody understand? You understand? Please say yes,
No,
thumbs up.
ma.
Let me,
No,
pardon?
ma.
No, ma.
No, ma.
No, ma, no, ma.
Okay,
I'll do now.
okay, okay. You know, proteins are made up of amino acids, and your amino acids belong to different groups. They are acidic, they are polar, they are non-polar, etc. You get that part. When you get your blast results frequently, they have a display. They'll tell you percentage aligned. Let's take this one, 100. Query 1 to 76. Target 1 to 76. Percentage identity 90.79. Percentage similarity 92.11. How did they get this? So just like you may find identity 100%, similarity 100%. Sometimes they are the same, sometimes they are not the same. Sometimes they are the same value, like the example we just did, 98.84. Sometimes they are not. So this is still from Ikena's file. I want you to look through these. The more we do, and I'll go back to that first one. Let's do another one. This one is also easy. Tell me, what do you notice about this? Tell me what you notice. Are there any differences?
Yes.
What do you notice? Tell me everywhere you notice a difference.
There is a place for D and A and B body.
Wonderful. So let's go. One thing I need to tell you is that these are written in 10. So this is 10, 20, 30, 40, 50. Please remember that. If we look at the first 10, once you see a single line like a tally, it means they match. MAVTVYDRNG. Can you see it? They're all the same. Can you see that? First 10, are we good?
Yes, ma'am.
Let's come to the next 10. Position number 11. Who agrees with me that this is position number 11?
Yes, ma'am. Yes, ma'am.
What do you see?
No.
What do you
Plus
notice
sign.
there?
Plus
You saw
sign.
what?
The plus sign.
A plus. The reason you saw a plus, what is D? What is represented by D? Aspartic.
Aspartic acid.
Plus
What is represented by E?
Glutamine.
Are they in the same group?
No, ma'am.
Yes, they are. They are. They are both acids. They have acid structure, so they are in the same group. So every other thing is the same, right? 12, 13, 14, 15, 16, 17, 18, 19, 20. Is that correct?
yes,
Correct?
ma'am.
I want you to look at 21, 22, 23, 24, 25. What is G? Glycine. What is D? Aspartic acid. Are they the same category? No. Glycine is polar. This is an acid. Do you see any line or plus sign here?
No, ma'am.
When there is no similarity and they are totally different, there's no line, there's no plus, okay? I'm going on to 31, 32, 33, 34, 35, 36. What do you see? Tryptophan and glycine. Are they in the same group? No. There is no plus. They are not identical. They are not similar. Let's go again. There's nothing missing here. 51, 52, 53. What is A?
Alanine.
Alar and valine. They are not similar. Okay. Come again. Tryptophan and glycine, serine and phenylalanine. You can see there's no plus, there's no line saying they're identical or they are similar. Can you see H and Q, glutamine? What is H? Histidine. All right. So what? Pardon? So this is a very clear example showing you when to put a plus and showing you why there's a plus, even understanding, because automatically it can generate the plus or nothing for you. So now when there's a space, you know that it's not identical, it's not similar, they are different, okay? All right. I want you to read my notes here for you. The amino acid residues are different at the following positions. Position 11, query has D, aspartic acid. So the top is always the query. I forgot to mention that clearly. And the bottom is the target, what you hit in the computer. So the target has E, glutamic acid. They are similar because they have the same physicochemical properties and are acidic amino acids. That is why you see a plus sign between them. Then they are not identical at positions 35, 10, 20, 30, 35. We have a W and G. Position 46, 10, 20, 30, 40. Where did I say 46? Yeah, okay, at 46, 10, 20, 30, 40, 41, 42, 43, 44, 45, 46. 46 is okay. That's an error there. Forgive. They are fine at 46. They are not identical at 54. They are not identical at 67, 68, and 73. So now you can see the regions where they're identical, right? In only one place. And then sorry about putting 46, because 46 is not among. So they are not identical at positions 35, 54, 67, 68, and 73. So let's plug in these numbers. So stay with me now on the math. What is the length, LS, of the number of aligned residues with the same characteristics? So you count. So the alignment of those that have the same gives 70. Then where you have the length of sequence A, 76, the length of sequence B, 76, you plug it in, 92.11. That's how we got the answer. I'm going to give you one from key one. So it's because this is not your page and it's an automatic answer. So you practice that. It will not make sense until you practice that. I'm going to give you a homework. I'll do one example for each of you with key one and Sonia, and then you will do that. Okay? Is that fine? Let's make progress.
Yes, ma'am.
Understood?
Yes, ma'am.
Pardon? Let me see if there's another one apart from Peterson, Sikena sequence. Identity. We see why it's identical. Different. You also see why it's different. See if I have one more example here. You should do one for you with Sonia and Q1. I will do that. I promise to do that. Let's make progress. This is some bioinformatics about alignment algorithms. There's a dot plot method. There's dynamic programming. I want you to remember this name, Needleman-Wunsch algorithm. That's then the Smith Waterman. That's the one for dynamic programming as well. And just some simple examples. You don't need to know all of this, but this is important so that you understand. This important because it's related with your FASTA format. There's also an amino acid scoring matrix. I just want you to be able to recognize this concept, like the Blossom 62, which is the software for alignment of sequences, and the PAM. When you hear of PAM numbers, those refer to amino acid scoring sequences. I'm going to revise these notes. You're not going to get this exactly. You're going to get it in Sonia and Q1. So if you have a favorite gene, you can send to me, Sonia Group, if there's a gene you want me to use to explain identity or blast results, I'll use that particular gene to explain that to you. So let me move on to something else. So your assignment that I'm going to give you is going to be based on calculating identity and going to be based on calculating identity and sequence similarity. Okay, when are you going to be able to submit it? I don't even know when I'll be able to send it to you. How many days would you need? No one has answered. All right. No one is answering. How many days do you need? Or you don't want to do it? You want to just see it in an exam? What was she saying about sequence identity? How did she count? How many days do you need? If I send it to you, maybe tomorrow.
3 days, ma.
Okay. All right. So phage proteins, you must look at the glossary on PhagesDB. On the first day of class, I mentioned this to you. So let me see if we can go there. Phagesdb.org. I thought you all had downloaded the glossary. Had you? Yes or no? Let me share my screen. Okay. So PhagesDB is very important, and it's something I need you to look at. Sometimes I've asked about I've asked about what's a database, and I used to like the question about what is an actinobacteriophage database, how is this structured, talk about the architecture. Well, past questions help a lot in this. If you understand past questions, you are 50% through. So on the left side, you should see that they have recently added phages, recently modified, finished phages, etc. You can see there's home. There's a place for you to do. Remember, a good database must have an organized retrieval system where you can search. Ease of retrieval must be made available. On the right side, it tells you the different phages. These are the genus, the genera, and they tell you how many phages have been found related to each of these. Now, if we take Sonia, for example, these are frequent things that make students freak out over nothing. So, oh, I've not seen that before. You must be able to understand what happens on Sonia's landing page. Sonia. So on the landing page, the first thing you see is these clear things, mirror images are plaques, as I was describing, and this place is a lawn of bacterium, the fuzzy part. And you can see that there's BLAST, if you remember, that the PhagesDB website too has BLAST. You can run GeneMark. GeneMark is a software that identifies START, and you land and you see that there's a detailed information for Sonia. What is the isolation host? That means the bacterium it infects, and it's Mycobacterium smegmatis. Who found it, the year it was found, the location, then the university where the scientists were studying it is stated, what program, it is the CPhage program, Science Education Alliance, Phage Hunters Advancing Genomics and Evolutionary Science. It will ask you whether it's from an enriched source sample. It's either enriched or direct. Enrichment means you put some of the bacteria just to increase the chances of phage lysine. There's the isolation temperature is always stated. You have GPS coordinates. As I click on the map, you will see the exact location that this phage was found. So that's why the GPS coordinates are there. And why is another reason why the GPS coordinates are important? If that phage has very strong therapeutic potential, you want to go back to the same soil where the phage was found. The discovery notes the soil was found beside the stairs behind Hen's Chapel. The phage is named after myself, so there's a Sonia Bilumani, so he named the phage after him or herself. Then sequencing information. Is the sequencing complete? Yes. When was it completed? 2024. Who was the sequencing facility stated? What type of sequencing method? For those who have had projects with me, you can say one or two things on Illumina. You know Illumina HiSeq, but this is Illumina MySeq. The read type, single reads. The read length, 150 base pairs. What is the coverage? 1,390. What is the genome length? Many times in exam, when you ask students, what is the genome length? They even read it from the table, but they forget to put base pairs. So I do not know whether it's centimeters you are referring to or 20 sheep. So they never get the right point. Character of genome ends, it can be 3', sticky overhang, the overhang length always. When they are sticky, you see the number of bases, 6, 10, etc. It will tell you. GC content is very important. 63.7. FASTA file available. Yes, you download the FASTA file. Now I'm going to go to the FASTA file because it's very, very important. In exam, students see this FASTA file and they are throwing off balance. When you see this in an exam, this line is called the definition line of a FASTA file. It will tell you the name of the phage. It will tell you whether it's a partial or a complete sequence. It will tell you the size of the genome. So there are two ways. You see it on the definition line of the FASTA file, or you can see the size directly. Peter, please go ahead.
Sorry, ma'am. The porter asked us to leave the classes. They want to lock it.
Who is the porter there again? Who is the porter? So that we'll tell him that we're almost done. Is it Mr. Ojo or the lady? Who is it?
The woman, mom. The female
She's
porter.
come back. Okay, tell her I'm coming. That we're having a class. You should let us finish. I'm coming down. Yes. Oh,
She's here.
that's what the text said. Maybe they think I'm not there. Very, very important. Abiona, what's up? Your hand is up.
Victor.
All right. So for Pages DB, usually you'll find characterization. Under characterization, you can find cluster information, cluster A1, cluster life cycle template, all the other cluster members. If you click here, Abishoes, Abrogate, Acme, so you can see, click on it again, it disappears. The annotation institution is the University of Lagos. Another important thing is Cyphoviridae, the morphotype. It's either Podo, Viridae, Cypho, Viridae. Please do not forget that. It can be Podo, it can be Cypho, it can be Myoviridae. So this is 81 number of genes. It does not really mean it can be 81, but we'll delete, add, and still be 81. It can end up being 82, it can end up being 80. But our correct annotation guides us. Is it uploaded to GenBank? No. GenBank accession number, you know what an accession number is now? Alphabets and numerals, unique numbers, so that you can retrieve the sequence anytime from public databases. RefSeq is a curated type, and we do not have one for it. So until we annotate this page. So this is very critical information. Now I can go to the gene list and I take Sonia 3. When I click on Sonia under script 3, if I want you to look at this page with me, this page, if you look through many things, call it, can you see it's 333? It's a hypothetical protein. It does not have a name. Please pay attention. On this page, there's a FAM number. PHAM stands for famerator. So this is the unique number, 28549. The genome position, pay attention, 2264 to 2584. Remember, when we are calculating length, we include calculating from the start. Ordinarily, you will have said 2584 minus 2264. You will get 320. But remember, we're calculating length, so you add 1, and that 1 is the start. The relationship between the length of nucleotides and amino acids is a 1/3. But remember, the last set of codons for the amino acid is a stop codon, so you don't add it. If you divide 321 by 3, you will get 107. But because you do not count the stop codon, it's not there. Let's look at another gene. So you can have screen displays because they're wondering, how is she going to test us? So let's take this one that is a minor tail protein. So what does this mean? If we investigate it to be true, and it has a minor tail protein like acanine, it has a minor tail protein like anglerfish, and it says they belong to the same protein family, 15611. How many of you have famerator maps?
I do,
How
ma'am.
many? How many?
I have famerator map.
We have famerator maps. Okay, that's good. All right, let me come here. How many of us recognize what this is? These were the things I sent to you for tail assembly chaperone, right? The places that are similar, where there's a difference. Have you done that homework I gave you?
Yes.
Pardon?
Yes, ma'am. Friendship.
Have you solved it?
Yes, ma'am.
Oh, we had, okay, just give it, all right, just let me stop my screen share. There was some, okay, I was going to show you some very quick notes on, let me get my downloads. Has any of you needed to delete? One second for this. Let me minimize this. What? Yeah. One second, let me show this. Let me see. 7. One second, I'm trying to show you something. All right. I left here to show you the glossary. Does everybody know where the glossary is? Yes, no, on Pages DB. Please indicate if you don't know where the glossary is.
Resources, ma'am.
Okay, wonderful. So you need to look at the glossary because I can ask you any 5 or 6 parts from there to tell me what they do. All right, so let's come to this diagram. These are phage proteins from different mycobacteriophages. Lysine A is a phage protein, lysine B, choline, tape measure, major tail subunit, there's a portal protein, there's a capsid maturation protease, and a terminase large subunit. I want you to look at this. This is across A1 phages. So the lysine A of acanine is 477, boiler 438, different sizes. Tape measure 666, 1991, 1216. These are their proteins and the protein lengths, okay? Now, I want to tell you how the farm number operates. When you see this, come back here, lysine A, and you see numbers like this number here represents the farm number, and the 617 represents how many phages have this farm number. So 5446 is the farm number. 326 phages are in this farm. 3527 is the farm number of the capsid maturation protease of cash flow, and 41 members are here. That was a few years ago. It could have changed. Here, I just show you forward. So I asked a question. Acanine, the tape measure protein, is from 16570 to 18570, and it's forward. What is the length in nucleotides? What is the length of amino acids? The length in nucleotides will be 18570 minus 16570. But remember, the start is among, so make sure you add 1. Amino acids, you will divide by 3, but remember, the last one will not be calculated. So how do you calculate length? 18570 minus 16570, 2001 base pairs. You count nucleotides, 1, 2. So this is included. So it's not 2000 base pairs, but 2001. For the protein length, you calculate protein length by dividing nucleotide length by 3. Then you subtract the stop amino acid. So the protein length is 2001 divided by 3, 667. But because the last amino acid is a stop, it does not code, we take it out, and the protein length is actually 666. Okay? Here, if you look at this gene, you will see that automatically, without you calculating it, it gives you, phages DB will give you the length in base pairs, it gives you the length in amino acids to show that our calculation was correct. This is how they came about it. So in exam, you may see the two displays, you should be able to recognize it. If you counted the number of amino acids from here to here, you will get 666. And now these are sequences you are very, very used to. And so if I ask you, what do the first 3 amino acid sequences represent? What will your answer be? MAA, what does it represent? And you think I'll give you the point for methionine? Are you sure I'll give you the point? What do you think I will do?
Sorry, Ma, can you repeat the question again?
You said methionine, you heard me quite all right, but I asked a question. Do you think I will give you the point?
Okay, I think it will start for.
Pardon?
I think it will start for another time.
Yes, instead of saying methionine, you should tell me it's the start codon. Okay, I want to just take you through how to delete a gene on DNA master. There are certain times, please pay attention in your annotation. You will notice that certain genes, no other person, you just go to phages DB, you see that gene alone, or another phage with that same gene, only 2 of them. But you have to be sure that it's really an orphan gene. You know how you say an orphan, no father, no mother? That terminology is borrowed in phage grammar, phage genomics. So we call it an off-arm. But just saying that there's just only one gene with it on phages DB is not sufficient. So you have coding potential, you have to check the coding potential, you have to check frames, you have to check phages DB, etc. I want you to look at this in red. The coding potential for gene 1 has a poor P. It is not situated. It is not situated within the gene, okay? You can see how normally our coding potential, there's a weighted line. We don't have time for all of that. So this is frames window. I hope we can do this tomorrow. Please come, Salem, remind me. Let's be able to do that tomorrow. Chijindu, Chiamaka, what's up?
No, I can only see your download.
You can what?
I see your download.
Oh, you only see my downloads. I'm so sorry. Wow. Okay. Let me stop share and share now. Sorry. Thank you for alerting me. Okay. So if you see this was cut off farm no data. That means no other person. This farm, only one member. You have to investigate it. So if you come to DNA master and you see that there are no blast results for this feature. So DNA master blasted, it did not give you anything. All the things I mentioned before, a weak coding potential, frames, then you go to phages DB, it says it's an off-arm gene, is an indication that you should be thinking of deleting it. So what you do is you click on the gene, see this arrow, and then you click delete. It will ask you, do you want to permanently delete this feature? You will say yes. And then it's going to delete the gene. However, you will want to renumber. So this is where you come to for the validation and you click these things, assign names, etc. This is very easy for you to click on and you should practice that tomorrow. Then you will see that there is a corrected gene. This automatically that is highlighted would be corrected. There will be renumbering, okay? So I was just showing you how to delete a gene. Now you know how to do that. Maybe not perfectly, but you know. Then another about the translational frame shifts. I want to show it again. You know, a frame shift is a mutation, very important. Of all the mutations I care about here is a frame shift. And it is caused by an addition or deletion of a base or base pairs in the DNA, and it allows a slippage in the translation of the genetic code. So it's very common with cluster A1 phages, and it happens in the tail assembly chaperone gene. You recognize it by 5 Gs. The addition is the 4th G, so take note. This will make the gene to be split into 2, which we have shown you. And what you can do, you can fix the gene by combining the 2 genes at the point that they split. So this shows you the first part, the CDS, the 5' end, the 3' end, and this second shows you, this is the 22, and this is the 23, okay? Now, when you look at the 22, it's always a perfect match, the first one. When you look at the second, it will still pull out tail assembly chaperone. That's why you know it's split. But then the query and the target, they'll just be little small pockets, okay? So I take pains to explain this. I'm not going to explain it as detailed. So you'll get these notes. So the same way Ayo and I were showing you template, complementary, you click to do that. It's 3 frames forward, how to read the things on top, how to read the ones below and reverse, okay? You've seen this before. These are just the notes. How we come back here. I hope you can see my screen.
No, ma.
You are not saying it. I am not talking. Baby, don't go. I've almost finished. 5 minutes. Why didn't you talk? You're just happy to say, let her talk, let her go. Emanuela, was that why your hand was up?
Yes,
All
ma'am.
right.
Yes, ma'am. We can see your screen.
Oh, thank you. Fixing frame shift. We already said these were the G's. The fourth G is the problem. In this example, we show you how to fix it in Gene 22 and in 23. If you look at the blast results, you see 100, 1 to 128, perfect match. The second part of the gene, 15 to 45, 237 to 267. So we come here, we show you genome, 6-frame translation. Then you come here, you've seen this before. We show you how to read it in frames, R, E, R. Each of these nucleotides will point to that. And we show you the key areas. Then we tell you where to search from. And you, after locating the frame shift, it is fixed. Then you come back to change the start of the second gene. Remember, you have 15915 to 16717. If you look here, when you first saw it, it was 16580. So you come back after you have noted where the stop sign was that caused the slippage. You come back to fix it. This slide show you exactly how you fix it, and you do region 1. You state the length, and then you validate. So this slide, these are things that we have to do in person tomorrow. We are 3 minutes to 8 o'clock. Good job, everybody. But I want to tell you, you need to hold on. You need to be very, very strong. You are almost there. To wrap this up, tomorrow we'll do online, we'll do physical, and then you should be strongly ready for exams. So please, I appeal to you. Let's leave that room as neatly. Let's not destroy anything. Pick up every liter, every grain of rice, every spoon, every plastic. Pick all your belongings, your phones, your chargers. We thank our graduate teaching assistants. God bless them. And I think we can call it a day. I will expect Joshua to get back to me on the agreed times online and in person. If you are going home, be safe. If you're on campus, try and get some rest. Drink water. But remember, these are very critical exams. Try not to play too hard. Rest a little and get back to work once you are revived. Thank you very much for your attention and participating. And those who are going home, be safe as you go. Good night.
Thank you,
ma'am.
ma'am.
Good night, ma'am. Thank you, ma'am.